Hi. When you deal with the bed files, which are indicators of peaks in your genome, of course you would like to count them all, and then if you compare different conditions, you would like to know how many of those peaks are in common and how many are different. And of course you think about something simple like a Venn diagram or Euler. And previously I told you that of course you can use bed tools to do some simple math with the bed files. However, bed tools do not give you a ready to use solution. You have to try hard to figure out how to combine different operators and get what you want. Instead, you can use ChIPpeakAnno. If you scroll through the tutorial, then pretty soon you come across Euler diagrams, which we will try to reproduce in this video using our data. In this exercise, I will use the same dataset as before, publicly available, where I previously generated average bed files. Details do not matter much, you just need two bed files with a minimum number of columns 3, chromosome, start and end position of the peak. That's enough. Although you can take a version of 5 columns, it doesn't matter much. Next, I compiled this pretty small script, only 44 lines. It's all based on the tutorial ChIPpeakAnno, and then I spent some time annotating and extracting gene names. Otherwise, what's the point? So what we do, we basically install ChIPpeakAnno first, and then we call it. Actually, you should not make any mistakes. Then, as I said, I take this public data, which are two groups of cells, IMOBs. These are primitive cells, and then upon some factors, it differentiates into neurons, all in vitro, mouse. These warnings you can ignore. Then pay attention that you do not use read CSV, you use immediately this special function reading bed files, which converts it to the G-ranges type. Otherwise, it doesn't understand, and you indicate the format. And then there are two lines. This is old-fashioned, it works, but then it will complain that it's deprecated, and therefore use this new name overlaps of peaks. It's a new one. This is goes to history. And I put max gap, so distance between peaks 50 nucleotides. I decided that if it's less than 50, then it's close enough and we can call it as one peak. Alright, we found overlapping peaks and we add colors, and then the step number 1 is already done. Even though it's called make Venn diagram, it draws Euler. Those ovals are not equal. And now the question is, can we get those peaks all in separate? And the answer is yes. So first of all, you ask what OL contains. It says you have overlapping peaks and you have unique peaks. And therefore you say common is OL $overlapping peaks. And then you can also ask for features, all details, how and where the overlap, and then you say, okay, how about unique? For neurons, this one for instance, it is not in the unique peaks, it's in a peak list neurons. That's how it works. You can also say OL peak list, and then dollar, and then you see that you have 3 options: neurons, IMOPs, or overlap. That means overlap. Okay, and so we get this new unique peaks this way, and then we can count in strength, which is either can be theoretically plus minus or dot. As I told you, dot is a problematic symbol in R. Very often it is replaced, and now again it was dot, now it is this snowflake. And 3215, it's differs from this number by 1. Where is one peak gone? I don't know. Alright, what is this unique neuer? It's of class G ranges, which is not yet the answer to our question. What we have to do, we have to annotate it at gene names, distances, etc. We do it this way. If you have a Homo sapiens, you have to use those two databases. It's all annotation databases of different kinds. These data are mouse. That's the latest number what I could find in a bioconductor. So I call this and then this. And it works in 2 steps. We call the libraries, and then we call the first library. Something was not converted. Okay, we have a little mishaps all the time. And then we select names out of unique neurons and call an annotation data from this database. And this is the first step of the annotation. If we type select it, then it is a little bit more to see. Then you see that these Ensmus gene numbers appeared. Compare with this unique neuron, chromosome, these strange X numbers, and that's it, nothing else. So step number 1 is already good progress. We have ensemble numbers. Now we call second library, annotation library, which has ensemble, entries ID, ensemble, protein, transcripts, gene names, etc. And therefore we call subset of it anno select out of this database selected feature. You can find this column over here, feature Ensmus gene number. It's actually double here and here. That's by these keys, key type ensemble, and what we need is a symbol. It's a gene name actually. Done. And then we merge. Merge is a common function in R. We merge annotation with selected table. And then we ask what it is and what it contains. There is one problematic column, merged column number 8, not just a list but composite picture. You can also open here and see that the most of the table is okay. This column will cause you trouble if you will try to save this data frame as it is. And we can sacrifice it, saying we don't need it. The rest is nice. Ensemble number, symbol of a gene, position, width. Don't be surprised by the variable width. It's not original max 3 bed file. It is averaged. Width changed during averaging and became variable. But again, this detail doesn't matter much. The focus of this exercise is to get unique and common peaks annotated by gene names and positions to those genes. And now what we want to save is all excluding number 8, right, done. Then we can open this saved table in a spreadsheet. I like LibreOffice. And then you can see that it's all nice and easy to understand. Peaks are now annotated by the nearby genes. The distances are indicated. Distance to a feature, it tells inside, upstream, outside. You can do pie chart out of these statistics, and so on. Using this table, you can select some locus and have a look at it on IGV. But I do not want to bother you with the details of this experiment. To summarize, if you want to compare two bed files and to find peaks which are common or unique for any of those bed files, then you use a ChIPpeakAnno and write an R script where by calling make Venn diagram you get Euler and the lists belonging to any of those groups. And in step number 2, you annotate those lists with the regular annotation databases, and you save your results and continue your study in any direction you like. That's how it works.